Thank you for attending our first virtual Marine Science Day. We're excited to have folks from all over the world with us today. My name's Jason Pafford. I'm a volunteer at VEMS, and I will be your host for this session. In this session, we're excited to have Dr. Eric Hilton, Curator of Fishes and Professor at VEMS, and Dr. Sarah Huber, Curatorial Associate and Collection Manager at VEMS, with us to take us on a tour of the Nunnally Ichthyology Collection. Hi there, Dr. Hilton.
Hello.
We've got lots of great questions for you, and I'll try to fit as many as I can in before the end of the session. Is there a most wanted list of specimens that you're wanting to add to the collection?
Ooh, that is a good question. Yeah, as you can imagine, specimens come from all over and in lots of different ways. And we have a list that we send out with some of our research cruises here at VEMS that we say, if we haven't collected any of these or preserved any of these species in a while, or could you, if you happen to catch some, could you please retain them? So those lists are built from what the surveys typically catch and what they have caught in the past, however long the survey's been going on. But we don't have necessarily a list that we send out to members of the public, which is another source of specimens that we actually have people come in that have caught a fish that they can't identify, or send us a picture and say, I've been fishing in this particular area for years and years, but never have seen this species. Can you identify it? So we do that. And once in a while, it's a unique record. So, we've had some people catch, I'm trying to think of a good example, but some stargazers, these fish that live in the bottom of the Chesapeake Bay and coastal regions of the mid-Atlantic. They look at, or they are relatively common, but they are not often encountered. And so we have some specimens that some fishermen caught a while back, and we're quite amazed that they've been living side by side with them for their whole life. Those are really interesting fishes in that they actually will bury themselves in the sediment, and then as a fish comes over the top, it will send out an electric pulse and stun the fish above it to secure its prey. So we get to introduce people into these really, really interesting aspects of the natural history of fishes as well.
And this all kind of rolls into another very popular question that we've gotten about the collection of fish. Are the fish caught live and then stored in the collection, or how does that come about?
Well, in various ways. So so for some species, some of our surveys during capture, those fish will die, or will, if we are expressly looking for a certain species, we will euthanize the fish. But a lot of our collection is opportunistic in that we don't want to see fish killed and then not used. So we preserve them and give them immortality, I like to think of it. For instance, 99%, and I might be overspeaking that, but a lot of the sharks from the shark longline survey that Sarah mentioned in her video, those fish are released alive and will live out their life. And that survey program is a really incredibly important metric of the shark population health along the coast. Once in a while, those fish do succumb on the long line. And an example of that is our white shark. The survey has caught several white sharks over the years. Most of those have been returned alive and healthy and have been tracked and live out their life. If it happens that that fish dies, we want to make as much use of it as possible. And like Sarah said, because of the size of the animal, typically people only preserve parts. So we can get a lot of interesting data, anatomical, ecological, even data off of, for instance, just shark jaws. So a lot of collections will only preserve the jaws of particularly large sharks. So it's very unique to have an entire animal that is preserved, that we can go back and look at all of the soft tissues of that fish, all of the guts contents, all of those aspects of the fish that are otherwise lost. So that is, that fish, even though it unfortunately didn't make it through the collection process, it has advanced the science and understanding of white sharks generally.
And naturally, the great white shark is a big interest. Do
Absolutely.
you keep that great white shark in a very large tank? What is the size of that specimen?
Well, so that's a really great question, and it does show the breadth of our collection generally. If we were at VIMS, we could show you that most of our specimens are in tiny jars and vials, no bigger than two inches, and that comes from our larval fish collection, which is estimated at about 50,000 vials of fish. But obviously you can't keep a 400-pound or 300-pound great white in a vial. We do have a bank, a storeroom of large stainless steel tanks that are filled with alcohol, the largest of which are about 8 feet long and preserve sharks, rays, billfish, large sturgeon, and so forth. So we have that breadth of storage capacity, from going from vials, most in vials and jars, then some smaller tanks about 3 feet long, and then about we have about 45 or so of those 35-gallon tanks, and then about 10 or 12 of the 300 to 500-gallon tanks.
Wow. Awesome. We do have a really popular question of how do you conduct DNA testing on fish? And what kind of fascinating information have you learned from DNA from fish?
Well, so my own research program, I'm just starting to dabble in genetics, mostly with the help of students here at VIMS and colleagues in the fisheries genetics program here at VIMS. But so before preservation of the specimen, we take a small muscle sample or trim a little bit of a bit of fin ray off of the fish and preserve that in 95% alcohol. A little tube, about 2 milliliters of alcohol. And that tube is then stored in at minus 80 degrees centigrade in a freezer, and that preserves and stabilizes the DNA for the long term. And then after that, we can preserve the fish, the actual specimen that tissue sample was extracted from. We preserve that in formalin, so researchers can go back and understand, wait, this fish that I sequenced the genetics from, and it didn't really fit with everything else, is actually a different species. And so one of the exciting things that in the last number of years that at VIMS, a graduate student, Christine Parsons, had a project on butterfly rays. And the butterfly rays from the East Coast, Gulf of Mexico, and the South Atlantic, they all looked similar, but she was finding a lot of differences in some standard fisheries data, age and growth information, so ecological type information. And the more she looked at the specimens that she was dissecting, the gut contents and age structures, she noticed some differences in the morphology of those fishes, different color patterns and so forth. She took genetic samples, those tissue samples, and sequenced those, and indeed there is a third species in that mix. So we're in the process of writing that up as describing this new species. Not new, it's undescribed species. So we're just starting to recognize that variation in that group of fishes.
Oh, that is awesome. Unfortunately, it looks like our time's just about over. Dr. Hilton, thank you so much for being here and for helping us learn more about that world-class collection of preserved fishes that you have there. It's been a pleasure chatting with you.
Wonderful. Thank you very much.
Thank you. For our attendees around the world, please navigate back to the agenda page to access our other live sessions. You can also browse the extensive collection of resources on the home screen. And we want to thank First Advantage Federal Credit Union for supporting this session. You guys have a wonderful day. Thank you so much.